and we'll try and get through them all as we go through the talk or at the end. And just to reinforce what Gail said, the lecture and the chat are being recorded, so make sure anything you want to say, you want to be captured. Okay, so I'm an organic chemist. I'm head of the Department of Chemistry. It's a great pleasure to be here. What I want to talk to you about today is how chemistry underpins really all the activity that goes on in the brain and therefore everything in the body. There are people who will tell you biology is the thing that runs the body. In fact, it's chemistry, so you just have to get used to that and accept it. Chemistry is the most important discipline for a variety of reasons. What I'm going to talk about today is how electrical signals are potentiated, that means made bigger or smaller, by chemicals, by small chemical molecules. And I'm going to talk about how the shape of those molecules is really important for the changes in electrical voltage that pass through the nervous system, both inside the brain and outside the brain, because the messages need to get outside. Those of you who can see my office, if you think it's untidy, behave yourselves. I just spent an hour tidying it. This is as good as it gets. Also, you need to know that I speak fast. Luckily, I don't have any sort of accent, so that's good, but you'll need to keep up. And the recording might be useful to you later on for that basis. Okay, so the brain controls the nervous system. And here is a cartoon showing the vertebrate nervous system. And within the brain and the nervous system, neurons are the core components of everything that takes place. And there are two subdivisions to the nervous system present in vertebrates. So a vertebrate is something that has a spinal cord or a spinal column, of which humans are one of the mammals which fulfill this criteria. Two branches, the central nervous system, CNS, which is the brain and the spinal cord, and the peripheral nervous system, which is essentially everything else. So the brain is at the top of the chain. The spinal cord is the key focal point where these messages are transmitted from the brain to the sites of action. So that could be muscles, that could be heart muscles, that could be muscles in your stomach, it could be muscles in your legs. So the peripheral nervous system is where the messages from the brain are turned into physical or chemical actions. Two broad types of neurons. Sensory neurons send messages from the body to the CNS. So if you feel a pin in your finger, the sensory neuron will send a message to the CNS and the brain will react accordingly. And motor neurons send messages to the body from the CNS. You may be aware of motor neuron disease, which is a very debilitating condition where motor neurons stop working, and so the ability to move and also carry out fundamental processes such as breathing and eating become more and more difficult. Research in our group is focused on issues to do with neurotransmission, including motor neuron disease, and this is why we're interested in this area. So what I want to do is give you an overview of the chemistry that underpins the biology of the system, and then for the last part of the talk, we'll talk a little bit about the chemistry of the system. So this is what a neuron looks like. It's a complex thing. When you look at almost all organelles present in mammalian systems, they are extremely complex. I don't expect you to pick all this up at once, but what I want you to do is to look in the circular boxes. So if you look at the top of the slide, I use my cursor to indicate what I'm talking about, show you a synapse. So a synapse is the end of a nerve cell that comes close to but doesn't touch because there's a gap, as I'm showing with the cursor here, another nerve cell. And that's how we're going to pass the electrical signal along. And these small little green dots, which are neurotransmitters, are the means by which we potentiate the electrical signal. Potentiate means turning up or down. So a volume control is a potentiator. It changes the magnitude of a signal for which is transmitted. So neuronal cells have all the typical mammalian cells would have. So they have a nucleus and nucleolus, they have membranes, microtubules, mitochondria, endoplasmic reticulum for protein transport, ribosomes for transfer of genetic material into protein product, Golgi apparatus, all these sorts of things. The key differentials is that nerve cells can maintain an electrical voltage. Now that is unusual. Most cells do not have anything other than a microscopic difference in electrical potential. Nerve cells can maintain an electrical charge. Now, when you bear in mind that everything in the body is in water, this is remarkable. How do neurons work without electrocuting the body? Well, if we look at these blue transparent sheaths known as Schwann cells, these are myelin. These are essentially fats which coat the nerve cell and insulate, just like a cable would insulate going from your TV to the wall. The metal which transmits the electrical signal to your TV is wrapped in a layer which is conductive, and therefore the electrical signal is retained. And the myelin sheath, which is what gets damaged in multiple sclerosis by the immune system, and that's why neuronal systems stop working with multiple sclerosis, that myelin sheath protects the electrical signal. Okay, so a nerve cell is simply a cell which can be electrically excitable. Now, what that means is not that it spills drinks because it's so excited. It means that it can be stimulated and can conduct electricity. That's how information is transmitted by the brain using electrical signals, but chemical signals can be used to stop or start those. And I've just made references to neurotransmitters, which I'll talk about again later on in the talk. The signals between neurons occur over those gaps via synapses. So the gap between the synapse and the cell to which the message is being transmitted is, as you might expect, called a synaptic gap. And there are many other specialized connections of neurons to other cells, especially where we're transmitting a message into an action, such as a muscle action. So neurons are the core components of the two parts of the central nervous system. I just mentioned those, CNS and PNS. Very highly specialized cells. Neurons don't look like any other cells. Three main types. Sensory neurons respond to stimuli received. I mentioned about a pin in the finger. The sensory neurons will send a message to your brain, and that could be touch, it could be sound, it could be light. Motor neurons send signals the other way. They receive instructions from the brain or spinal cord and cause muscle contractions and also glandular outputs and control the rate of heartbeats, lung interactions, that sort of thing. The third class, interneurons, connect neurons to other neurons. That's normally within the same region of the brain or the spinal cord. Three key structures present in neurons: the cell body, which is known as a soma, dendrites, and the axon. Here's a simpler version of this, the drawing that I just showed you, which homes in on the key features. So the dendrites, named after dendrimer, where we have multiple divisions at the end points of the branches. So it looks kind of like the branch of a tree, a very complex, a very densely structured region. The nucleus, which is present in all mammalian cells, except red blood cells, where it's removed during the processing. The cell body, which attaches to the axon. And then the myelin sheath, as I mentioned, the Schwann cells. And those gaps in between, known as the nodes of Ranvier, they are also important for the overall activity of the nerve. Now, the signal is going to be sent along the axon via the axon terminal, and that's how the electricity will flow in the brain. There's a more glamorous version of exactly the same arrangement, just shown in a little more detail now. So the synaptic terminal is shown at the right. These are the things I just showed you, the end of the neuron where there's a gap. And by either flooding or turning off the chemical signals that flow across the synaptic gap, we can turn on or off the electrical signal, and we can make the magnitude large or small. And so that's a really important feature. So that's our volume control for the neuronal signal. Okay, so neurotransmission is the flow along the neuron from the axon hill towards the synapse, which I'm tracing out here with my cursor. The electrical signal is known as the action potential. And the way we generate electricity in the body without setting fire to ourselves, with the equivalent of dropping a toaster into the bath, is by moving ions in and out of the neuronal cell. Now, if we take an ion, a positively or negatively charged thing, and we remove it from a charge balance situation, we create a charge. If we create a charge next to an opposite charge, that leads to a force, and that is an electrical current which can flow. Three broad types of stimuli can induce electrical activity: physical, electrical, or chemical. I mentioned about neurotransmitters being the small molecules that are released. I'm showing at the diagram at right here how they're released across the synaptic gap to transmit the signal. Okay, this is a really complex slide. This is recorded so you can look at it later on. The things you really need to know are shown at right. So normally the so-called resting potential, we have a high concentration of sodium ions outside the nerve cell and a high concentration of potassium ions inside the nerve cell. Now, sodium and potassium ions are really important for neurotransmission. That's why you should eat bananas after exercise, because bananas are rich in potassium. And that's why high salt food can have an effect on neurotransmission, because sodium chloride is, of course, salt. So that's the way it is normally. When there's an action potential, what we do is we change from an even positive on the outside of the cell. Most cells have a positive charge, very slight positive charge on the outside. A resting potential that is even all the way along. The action potential occurs when sodium goes against the concentration flow into the cell, and that creates a region of negative charge because we've taken positive charge away from the outside. When we put a positive next to a negative, there's an attractive influence, and that leads to the electrical force which transmits the signal. Now that potential, the difference in electrical situation between the inside and the outside of the cell is known as the membrane potential. And that changes due to the membrane permeability. Permeability is the ease with which something goes across something. And so ions moving across the membrane can change the electrical balance, and that changes the membrane potential. So the potential, you know about potential. So batteries have a voltage. A 9-volt battery has a potential of 9 volts. There are different sorts of potentials. There can be short-lived local changes, and they give a signal over a short distance, and they're known as graded potentials. Now, the ones that we're really interested in this talk are long-distance effects, and they're known as action potentials. Two other features you need to know about: when you reduce the membrane potential, in other words, the inside becomes less negative, that is a depolarization. The opposite is a hyperpolarization. Now, they're really important, for instance, in heart arrhythmias. The length of time for the depolarization and the hyperpolarization are intimately related to diseases of the heart. Okay, so for graded potentials, what happens? So the stimulus causes channels to open. We'll see in a moment that membranes aren't solid things. They're not like balloons. They have holes in them. Unlike balloons, they can maintain their shape even when those holes are open. And the way we get ions into a cell is by moving them through so-called channels. And a gated channel is something which is not always open, but can be open and closed by a signal. That can either be electrical or it can be chemical. Okay, so when ions move across the membrane, that gives that small region a negative charge when sodium moves in, that I just showed you. Then ions move along to neutralize that charge difference. Inside the membrane, potassium ions move away from the active regions to adjacent ones, and that pushes negative ions away. Outside the membrane, potassium ions move towards the active regions. That means that those regions become less positive, and also areas around become depolarized. As usual, a diagram summarizes 1,000 words or more. This is what we're looking at. So here is the negative region caused by sodium moving into the nerve cell. And that leads to a force where the positive is attracted to the negative charge. Outside, the potassium ions move towards the active region. So this is the active region. The positive charges move towards the negative charge. Of course, on the inside, we've now got an excess of positive charge from sodium ions. And so the potassium ions move away. Positive repels positive, like the poles of a magnet. The similar poles repel each other. And that force is what moves the ions away. And the movement of charge is what defines an electrical signal. Okay, how do we get these ions in and out? Okay, we have to have gates to prevent the cell from losing its structure. Ion channels, which are the mechanisms by which we get ions into and out of cells, are proteins, and they are what move ions across. Now, there are some that are always open, and so these slowly leak ions in or out, and they're called passive channels. The gated or active channels open or close in response to chemical signals. So chemically gated channels open when a neurotransmitter binds. Voltage gated channels open in response to change in the membrane potential. Okay, I mentioned that membranes are all polarized. Normally, it's a tiny polarization. In neurons, it's about -40 to -90 millivolts, so thousands of volts. That is a colossal charge for a system which is covered in water. Now, that difference in electrical potential exists only across the membrane. Inside and out the cell, there is an overall neutral charge. And it arises from what I just showed you, the motion of ions. So here's a cartoon again. 1,000 words summarized, at least in this diagram. So this rather fetching pink purple color is the active channel, which is known as the sodium potassium pump. And then the kind of teal aqua ones, these are passive channels that are always open. Now, in the passive channels, we go from a high concentration region, so potassium has a high concentration inside the cell, to the lower concentration region. That's a normal thermodynamic effect. And the same on the other side for sodium coming into the cell. So the two passive channels will be permanently open and slowly allow excess potassium to get outside and excess sodium to get inside the nerve cell. The key here, though, is the sodium potassium pump, because that pushes sodium to a high concentration and potassium to a high potassium concentration in the cell. And so that requires energy because it's going against the normal concentration. We have to put energy in to go against the repulsive force of moving towards high concentration. And this is an active channel. It can only be used in combined with energy. Okay, the two types we have, chemically gated, we have a closed channel here. Here is a little notch on the surface, and that is where our neurotransmitter binds. When a neurotransmitter comes in, it pushes out the structure of the protein and opens up the channel, and that's where we allow the ion flow. For a voltage-gated system, changes in the membrane can also change from a closed to an open system. We see this little flap, which is opened up in this case. And so when there's an electrical change by changes in charge in the membrane, that can also lead to the movement of ions across the membrane. And we're going to talk in a little bit about neurotransmitters and how they chemically gate ion channels. Okay, so potassium steadily diffuses out of the cell with the concentration and gradient. Sodium does the same, but the opposite way around. The voltage is maintained, that is, the potential is maintained by the sodium-potassium pump, and it's a fascinating protein which is evolved to transport 3 sodiums out of a cell for every 2 potassiums brought into the cell. So that means that we're 1 short in terms of positive charge. 3 positives out, 2 come in. Okay, so that's an important thing. And this is a little GIF which hopefully will show you how that works. So here is the protein. Down at bottom right, this is a neurotransmitter, and we'll see that when this is present, it has one action, that is the ion channel has one action. When the transmitter is not present, it has a different one. So we've got 3 sodiums bonded there in the sodium-potassium pump. And what happens is there's a change in the shape of the protein. We dump out 3 sodiums, we bring in 2 potassiums, and notice the difference. When the neurotransmitter is off, it's closed on the outside. When the neurotransmitter is on, it's open. And so that neurotransmitter gates the input or output of ions. And that's how neurotransmitters, chemical structures, can control electrical signals. And here is how the action potential moves along. Here's a system, a resting potential, where we have a balance of positive and negative charges. And then as we move sodiums away, we generate negative charges. That leads to a force. And that negative charge moves along the axon, and the electrical signal goes through the terminus of the neuron and over the synaptic gap. So by moving ions, we can create electricity at a small level, but enough to have major physiological effects. Of course, there are also psychological effects. Okay, here is the system I just shown you. This is the same slide, the vertebrate nervous system, 2 sorts I mentioned, sensory and motor neurons. And within the nervous system, there are 2 different parts. The autonomic nervous system, also known as the visceral nervous system, and this controls unconscious things, things that we don't want to have to think about. So we don't want to have to remember to beat our heart. We don't want to have to remember to digest food. We don't want to have to remember to breathe or to keep our eyes open and other less pleasant things. And so the autonomic nervous system controls all of these things that go on in the background all the time without us knowing about it. And within the ANS, the autonomic nervous system, there are 2 subdivisions: a sympathetic nervous system known as the fight or flight system, you've probably heard of, the parasympathetic nervous system, which is known as the rest or digest, and less pleasantly, the feed or breathe system. Okay, it's a complex system. On the left, we have the sympathetic autonomic pathway. On the right, we have the parasympathetic. No need to dwell on the detail. Just notice what is controlled here: blood vessels, thyroid gland, lungs, heart, liver, kidney, stomach, other parts of the body that are important, but we'll just pass over for the moment. The somatic nervous system, on the other hand, is the voluntary nervous system, and that's associated with the things that we do want to control with our thoughts and our brain impulses, such as skeletal muscle movements. So in other words, anything voluntary, when we want to move our hands around, as I do because I'm overexcited, our brain does that through the somatic nervous system. And there's 3 main parts: the spinal nerves, the 31 segments of nerves and spinal cords, the peripheral nerves carrying sensory information, and motor commands into and out of the spinal cord. There are also brain nerves, specialist brain nerves known as cranial nerves, 12 of those, 12 segments within the brainstem. They carry the information into the brainstem, view of the brain. Without the brainstem, the brain cannot work. And then there's a so-called association nerves. There are thousands of these nerves, and they pull together the sensory input and the motor output that you need to be able to do things under voluntary control. Okay, why is this important? Well, I'm going to show you just now how neurotransmitters interact with the autonomic nervous system on the left and the somatic nervous system on the right. So here are the automatic things we don't want to think about. Here are the motor motions that we do want to think about. Neurotransmitters act in these regions. So for the somatic nervous system at muscles, they act at the muscle to turn the muscle on or off, and in really precise ways. You know how well you can control your hands. These are very precise messages that are sent out. And in the autonomic system, then it's the same sort of thing, but for autonomous processes. Okay, so neurotransmitters, chemicals. Amino acids are the neurotransmitter molecules. Now, glycine is the simplest amino acid. I noticed that this resolution isn't great. I apologize for that. But glycine has one nitrogen atom and then two carbons. Now, we normally indicate carbon-carbon bonds as kinks in a line structure because it's just easier to avoid writing CHs all over the place. So here at the kink, we have a carbon and we miss out the hydrogens to make it easier to draw. So that's the chemical formula that we'd write down. This is what it actually looks like. So what we've got, you'll be familiar hopefully with the concept of pyramidal structures. So we have tetrahedral structures of these atoms. There's a blue nitrogen atom. We have a lone pair of electrons pointing out into space, which we don't normally represent. The plane of that nitrogen lone pair neatly bisects the bond angle between the CH2, and that's a beautiful system. And we have this carboxylic acid. Oxygen is red, so a CO2H. Now, when you have an acid and an amine is a base, then you have an acid-base reaction. And so amino acids exist as Zwitterions in vivo in aqueous solution. So glycine is actually a positive-negative system. So rather than having the arrangement here with one hydrogen and oxygen and a double bond to oxygen on the second of the oxygen atoms, it exists in this position. So we switched around the hydrogen from oxygen to nitrogen. Nitrogen has become positively charged, and now it's symmetrical. It has three hydrogens and one substituent here. Notice the shape of this CO2 group has now changed. We no longer have a hydrogen, but we have a negative charge. And that's really important because negative charges are very different to neutral things. Negative charges will bind very well to positive charges, whereas neutral things don't bind at all. So the shape and the charge is really important. The primary mammalian neurotransmitter is glutamate, and that's a substituted amino acid. It has another group bound to it as well as hydrogens, and it has a very distinctive shape. So it's actually an amino diacid. We'd have two acid groups. So whereas in glycine we had two of these carbon-hydrogen bonds, we've got this additional acid group present in glutamate. And this is what glutamate looks like. I've shown a slightly different version now, which is not space filled. This doesn't show the electron density because it's simpler to draw it this way. Here are our oxygens present in the carboxylates. Here's our nitrogen with that pyramidal structure, the lone pair down here. And so you can see now that the shape of this molecule, there are many ways we could draw the conformation, but the shape is something that is really important. So that's glutamate. Notice that this hydrogen, carbon-hydrogen bond is pointing away from us as we look at it. Another main amino acid which is used in neurotransmission is N-methyl D-aspartate. Now that has the opposite shape to glutamate. It's got one less carbon in the chain. It's got a methyl group, a carbon atom present on nitrogen, but it's also got the hydrogen pointing towards us instead of away from us. So that means that it's got similar carboxylic acid binding groups to glutamate, but a very different shape. And so NMDA, which you may have heard of, will send very different signals to glutamate. So the shape of the molecule can precisely control the nature of the signal which is passed from brain to the tissues in the body. So NMDA is a key neurotransmitter. Okay, so neuroexcitatory amino acids, there are two main classes of compounds, so-called metabotropic or ionotropic. Metabotropic are named in this way because they change metabolic processes, so they are very slow. Metabolic processes are stepwise chemical reactions that make or break chemicals into or down to other molecules. Ionotropics are things that influence ion change, and ions move fast. So ionotropic amino acids are involved in very fast bodily effects. So some of you may have been unfortunate enough to have food poisoning. That is normally caused by amino acids. So amnesic shellfish poisoning is a classic version of the illness. It can cause vomiting, nausea, diarrhea, all the things you'll know about if you've had it. But also beyond the simple poisoning, you can also get very serious conditions. So headaches, dizziness, seizures, profuse respiratory secretions, unstable blood pressure. This kind of, if you're a Manchester United supporter, when Liverpool won the league earlier in the year. And so these effects can be really rather pronounced. Now, there's an interesting example out there. In Capitola, which is a small town in California in 1961, there was an episode where hundreds of seabirds dived into the seaside town, crying like children when they dived into street lamps, windows, and attacked people on the ground. And this story was the basis of the film The Birds, which Alfred Hitchcock made a couple of years later. So these amino acids have terrific effects. The effect on the birds I've just mentioned was caused by an amino acid called domoic acid, an ionotropic amino acid. And here's what it looks like. It looks quite complex. Don't worry about the structure. Just focus in on the yellow part, because that looks like glutamate. And so domoic acid is what we refer to as a glutamate analog. It acts at the same binding site of glutamate in neurotransmission, but it leads to very different physical and biological effects. And so these compounds, which are made in nature as defense chemicals by bacteria, for instance, or algal blooms, if they're eaten by birds, it will stop the birds from eating them again. These defense amino acids can have really pronounced physical properties. Okay, so shape's important in chemistry, but also in biology. So glutamate, I showed you, is bound at the glutamate receptor at proteins, and here's what it looks like. Don't worry about it, it's complex again. Beautiful corkscrew-shaped alpha helices here, alpha helix, the predominant motif found in pretty much all proteins. Down here in this little gold structure is our domoic acid. And you can see it's minuscule compared to the protein structure, and indeed this is a subsection of the protein, but it causes all the biological activity. And if we focus in on the part in the red box, we can see this beautiful right angle arrangement of the carboxylic acid groups here, here, and here. So one carboxylic acid, another one, and then another one, all mutually at right angles. And the binding of those negative charges in the carboxylates, because remember, I showed you that's a CO2 minus, and this is an NH2 plus. That negative charge allows this molecule to sit tightly in the protein and cause the effects. Now, we don't want to do things like this. We don't want to use these compounds to poison people. But by understanding how these compounds work, we might be able to make things which we could use, for instance, to treat motor neuron disease. So understanding how the shape of the chemicals is important for the biological effect is something that drives our research and the research of many thousands of groups around the world. Here is a slightly simpler molecule binding to a glutamate receptor. This shows the beautiful symmetry of these things. Again, we have the red alpha helix and then the flat beta sheet shown in this aquamarine color. The small molecule bound here is canic acid, which is a smaller analog of domoic acid, which has many of the effects of domoic acid and is used as a tool in the study of neurological conditions around the world by biologists. The only difference between domoic acid, and canic acid is this part of the molecule. It's just a little shorter. And you see again the right angle arrangement of the carboxylates, which is responsible for the binding activity. So understanding the shape and the charge in these amino acids, these small chemicals, is vitally important for being able to understand and therefore control and predict neurotransmission. Okay, so there's bunches of other neurotransmitter molecules. So acetylcholine, acetylcholinesterases are nerve poisons. Noradrenaline, you'll have heard of adrenaline and noradrenaline. Dopamine, which is involved in Parkinson's disease. Serotonin, which is involved in all sorts of conditions to do with mood and stimulation. Histamine, to do with allergic reactions. Adenosine. Adenosine analogs are found in coffee. So caffeine is an adenosine analog. That's why adults need coffee so much in the morning and to keep them up in the night. And there are many simple compounds. I've just shown you glycine with the, I apologize for this bond angle, it's not my drawing, aspartic acid and glutamic acid that I showed you. GABA, you'll have heard, is a notorious amino acid. And also peptides, so enkephalins, which are natural painkillers, and the enkephalin binding sites are where opioids such as heroin and morphine act. These are the natural painkillers that are released in the body whenever we have any sort of pain compromise. Substance P is a different type of peptide. This causes the sensation of pain. And remember, pain, as I was saying to a bright young man this morning, is something that lets us know that we're alive. So neurotransmitters, both charged and uncharged, notice we've got carboxylates at the end. That's what this OH means. So these are charged systems. We have an amino group, which is also going to be charged. These small compounds and bigger peptides control all of the things that we see in terms of the stimulation on our periphery and also the signals that our brain sends us. Okay, just finally now I've got a movie to show you exactly how this all comes together. And this, I think, summarizes how beautiful the nervous system is. So here's our brain. We swoop down with our I-SPY camera and we see these electrical signals moving around the neurons at really breakneck speed. This is slowed down about 1,000 times. Here are the ions going in and out of the neuronal cell. We're going to focus in a moment. There's the sodium ions going in and coming out, going in and coming out as the sodium potassium pump does its job. And that leads to the force, which is indicated by the sound here, and the electrical charge moving along to the synapse, where these small little spheres go to the gap and then release the small molecules, which you can see here, which lead to the opening and closing of the ion channels. And this is a slowed down version of that event. The vesicle comes into the synaptic endpoint. It releases its neurotransmitters. That opens up the ion channels to allow ions to proceed, and that sends or stops the chemical signal from being received. Okay, so hopefully you've got a feel now for how important chemistry is, the most important science, especially in terms of biology. The shapes of the molecules and the charges are really important and really can help us to understand how the brain works and how we can fix parts of the brain and the nervous system that don't work so well. Okay, thank you for your attention.